Okay, hello everybody. Welcome to our webinar Wednesday. I'm Monika Burba, Product Manager for Molecular Biology and Cell Biology products at Biozyme with more than 20 years experience. We are proud that so many people, customers and scientists, join our webinar today. Thank you very much. The aim of Biozyme webinars is to transport new and exciting information about laboratory technologies and new products to all interested people. Today, we will share comprehensive information with you why and how to switch from cell culturing in classical to serum-free media. I'm very happy that Nicole Wellens from Lonza today presents us better insights into this exciting theme. Nicole is Senior Product Manager for Cell Culture Media at Lonza Bioscience Solutions with more than 30 years of experience in cell culturing and is a real expert on serum-free cell culturing media. At the end of this session, we will have about 5 minutes for discussion. Please type in your question into the chat box whenever you like. We will answer them at the end of the session. Thank you in advance for your input. Nicole, please give us an insight into the serum-free culturing of cells.
Thank you, Monica, and welcome everybody to today's webinar about serum-free cell culturing. As Monica already mentioned, my name is Nicole Wellens, and I am the product manager for our cell culture media within the Lonza Bioscience division. Probably, as you know, Lonza provides life sciences with researchers with the tools they need to develop and test therapeutics from basic research to final products. During today's session, I will introduce you to serum-free cell culturing. I will start with an overview covering concerns when cultivating cells in medium containing fetal bovine serum, also known as fetal calf serum. And in cell culture, I think we're all familiar with batch-to-batch testing of serum, and often the real reason behind why we do this is forgotten. I will also talk about the different choices there are in serum-free media formulations, and I will guide you through the easy process of switching to serum-free cell culturing, and I will discuss what serum-free possibilities there are and show you some examples. Unfortunately, because normally this was a PowerPoint and now things are over each other, but I hope you can see it anyway, but I will tell you what this slide I will try to explain here. So we are all familiar with cell culturing in vitro, and the way of why we are doing this is to mimic in vivo conditions. And of course, fetal bovine serum is used as a universal growth supplement to culture cells in tissue, in media like classical media like RPMI and DMEM. And of course, FBNs contains your natural cocktail of growth factors, hormone, lipids, and minerals. And of course, we know these are required for cells to attach, to grow, to proliferate. And it's effective for most cell types. It's not, well, for human cells, animal cells, and also insect cells. Although serum has been in use for over 15 years, 50, it has never been fully characterized. The serum concentration we add can vary today between 5 and 10%, and sometimes I read even higher concentrations. And to be honest, I'm not really sure what happened with the cows or with the cells. When I was still working in the lab, 5% was the golden standard. Well, the current addition of serum to me seems very high. So now we identified why we use serum, I would like to make you aware about some concerns and future concerns of the use of serum. So when we talk about serum, we talk about an undefined product of which the exact composition is not known. There are not just one cow species. The diversity of cows worldwide is big. To give you an idea, there are over 1,000 breeds of cattle. So this will raise the question, what do we know about the serum we use in the lab today? So the large variability of serum results in batch and lot variation, which you are still familiar with, and this batch and lot variation can ultimately lead to experimental variability and limit interlaboratory reproducibility. We most of the time do not exactly know where the serum is originated from. What was the status of the cow when the blood was taken? Were they nervous? What did they eat the day before? And what the time point was it was taken? And also, is it a mixture of different cows or different factories? Based on all these questions, we can agree working with serum is working with a product whose composition is undefined and not fitting with exact scientific results. Of course, I can tell you that, but there are also many publications, and I just wanted to highlight one. This is one, well, it was published in 1982, so already a long time ago, by Price and Gregory, but the one I show you here is a referral to this one by a more recent publication by Mr. Gerhard Kristronthal, difficult word for me. And what Price and Gregory did, they tested the composition of different serum batches, and they found a broad concentration range per component tested. And if you look at the slide, so what you see at the right side, so here is the total, or the different components that they tested. The concentration is the average concentration of all the components tested in the different batches. And here on the right side, you see the range, so the lowest and the highest amount that was measured in a specific batch. And I think you can agree that there is quite some diversity and there is a broad spread. So this would mean also that the standardization of your work becomes challenging when using serum. And of course also costly if you're not able to find a new batch which works with your cells. Well, we identified serum being undefined components, which makes standardization difficult. And probably you never thought about fraud would also be part of the concern. In, I think it was 2013, PAA Laboratories sent out an urgent notification, safety notification, where it was mentioned that during 2003 and 2013, so over a 10-year period, the FBS may have contained adult bovine serum albumin of United States origin, water, cell growth promotion additives, and in addition, the country of origin stated on the label might be incorrect or a mixture. And this was not the first time fraud in relation to serum was reported, and I will tell you a little bit more about that when I talk about global demand. Also, fetal bovine serum used in cell culture is a byproduct of the meat industry. It is produced from blood collected at commercial slaughterhouses from pregnant cows. We often we live in is changing. Animal welfare is getting more and more important. Eating meat is still common, although not eating meat every day is more and more promoted. We hear daily in the news about global warming in relation to carbon dioxide emission, and animals are a major contributor here. So what consequences might change in human behavior for the future and is related to the availability of serum, meaning also the price will probably increase. And I think you can agree that we have already seen huge price increases the past decades. Sorry. One question to you. So are you familiar with the content of your certificate of analysis of your serum? So for instance, do you know if your serum is tested for mycoplasma? Because mycoplasma is an infection that cannot be seen by the eye, but it can have a major effect on the reliability, reproducibility, and also the consistency of your experimental results. They can even cause slower growth or even, cell death. So I will take something to drink. And you might, you might think that this was relevant in the past, but I have seen a recent publication from 2017 where they tested commercial batches And in this case, they did not test it for mycoplasma contamination, but also concerns are, besides mycoplasma, endotoxins, viruses, and also prion proteins. And in this case, they tested if there were viral traces present, and all commercial batches tested, with only one exception, had a lot of virus traces present. That might also raise a concern in this case. Last concern I would like to discuss. So if you're not concerned yet about what I told you about safety, fraud, or the effect of the undefined composition of serum, I think the availability of serum in the future should be your concern. What we know is that the global photo-bovine serum market was valued at, let's say, 700 million euro in 2017, and it's estimated to reach more than 1 million euro in 2025. So that means almost a doubling of the demand required for biopharmaceuticals, human and animal vaccine production, but of course also research. And we talk about millions of cows slaughtered each year. So what we can see that global demand is increasing, prices go up, supply is depending on slaughterhouses, meat production, and this depends again on farmers bringing their cows to the slaughterhouse. If the farmer, by any reason, decides to decrease the size of the cattle because of dry summer, bad weather, or because just of lower human consumption of meat, or even by a decision by politics, the serum availability is affected, and when products become scarce, prices go up. So an important question is, will we have enough to serve the demand in the future? We already spoke about fraud earlier, and I mentioned I would tell you about another example. This is a little bit more back in time, so it was related to 1994, and sold worldwide. However, at that time, only 50,000 liters of high-quality serum was annually collected in New Zealand. So there was a big question, how can we ever have sold 30,000 liters while we only collected 15,000 liters? Well, with all these concerns, you might not wonder why at least we see a huge interest in going serum-free or starting to culture your cells serum-free. So let's have a look at the serum-free alternative which are available in the market. So in general, if we talk about serum-free media, it means that the media does not contain serum and it does not require it to grow cells. And there are different types of serum-free media. Besides only being serum-free, there are also serum-free media on the market which is so-called protein-free. So that means that they do not contain proteins smaller than 10 kD, and they may contain some insulin, but it's only 5 kD. And serum-free, protein-free media might be, for instance, of interest if you would like to produce a protein, or if you would like to investigate the stimulation or inhibition of a certain protein. Serum-free media can be of non-animal origin. That means that there is nothing of animals, which includes us, humans, in the medium, and it can contain, for instance, some plant hydrolysates. The golden standard of serum-free media are the chemically defined media, because they contain only components of known structure, nothing of animal origin, and only recombinant proteins and no hydrolysates. And this is, as you can imagine, the most expensive serum-free media. And this is more and more requested by the industry to avoid any undefined material in the media. Probably you have seen that I skipped the xenon-free media, which means that it does not contain any proteins of a strange origin. So in case, I don't know if you're familiar with that, but Lonza has a product which is called ex vivo, which is dedicated for cell therapy, so culturing hematopoietic cells. And this medium only contains human or recombinant origin components. And in cell therapy, you can imagine xenon-free or chemically defined is often the choice of the media. Just to mention in addition, so it's not one or the other, so serum-free media can be a combination. For instance, they can be non-animal origin and protein-free, And of course, if we talk about chemically defined media, they are by origin already non-animal origin. So knowing about the choices in serum-free media, what considerations are important when moving to a serum-free media? So I already spoke about the concerns when using serum. So why is it a good idea to culture serum-free? So serum-free media can be of general purpose. Then you can really easily compare it what probably you're currently using, like RPMI with the addition of serum. On the other end, most serum-free media have a formulation specifically developed for a specific cell type. And of course, we would like in vitro to mimic as much the in vivo situation. And of course, the situation for each cell type is different. They do not have, like blood cells, direct access to serum. So there are media specific for dedicated cell types. And just to name a few, we have specific media for kidney cells like NDCK or HEC293. We have specific media for hematopoietic cells like T cells or dendritic cells. And I will tell you a little bit about that later. And we also have specific media for mammalian cells like Chinese hamster ovaria cells, and of course for insect cells like SF9. And the formulation for serum-free media is more standardized. So chemically defined media are exactly formulated the same with each produced batch. This removes the requirement for batch-to-batch testing. Serum-free media do not contain any serum, and as I discussed earlier, serum is often a source for mycoplasma contamination or virus contamination, which can have a huge effect on cell growth. And this is all avoided when going serum-free. Often thought that serum-free media are more expensive, and this all depends on what you're comparing. Serum-free media should only be compared with the highest quality of serum, and even then, serum-free media are higher in quality due to the standard formulation. No requirement of batch-to-batch testing and no concerns for safety. Specific for those customers who produce proteins, the yield of protein production is higher in serum-free media, and that is mainly related to the less intense downstream processing. So you have to do less steps to purify your protein, and as we all know, each additional step that we can avoid in downstream processing will give us a higher yield. So when starting with serum-free media, you have to consider your cells might need to adapt to the serum-free media. For this, you have to follow a process called weaning. This might take some weeks, depending on the proliferation speed of your cell line. Well, you have to compare it like putting your cells on a healthy diet. So there are weaning protocols available from Lonza, which takes you through all the steps, and I will explain you a little bit later about that as well. The cell growth rate might be affected, and in case of protein production, this might not influence the protein yield. A subpopulation of cells that can proliferate in the absence of serum might be selected, and this is not that different than when you work with undefined serum. But consequently, a serum-free media adapted cell line can be a little bit different from the original cell line in a serum supplemented medium. And when changing to serum-free media, you have to compare the price, because price is always a discussion, as I already explained. You have to really compare the price with a medium higher than the highest standard of medium supplemented with serum. And for protein production, look at the increased yield and the easier downstream processing. So what are the consequences of moving to serum-free environments? I already mentioned that cells have to get used to growing in a serum-free environment. And let's say that if they are in serum, they are at an all-you-can-eat menu. And if you move them to a serum-free, they're on a healthy diet. And there are different ways of doing that. So you can do a sequential adaptation, and there are two ways of doing that. The first one is to dilute the serum. So you immediately start with working in the serum-free media, but you still add the serum to it, and gradually you take off serum, so you go from 10% to 5% to 2.5%, and finally end up with 0% of serum. What is important if you do this weaning process that you take into account the viability. It always should be more than 85% before you lower to the next level of serum. And if it's not, then just keep your cells a little bit longer in this specific serum concentration, and then finally you will end up with no serum. Another way of doing it is the gradual dilution. And what you then do, you do a mixture of your classical serum media with your serum-free media, and also then gradually move away from the classical media with serum and add more serum-free media, and then add up with a fully serum-free of cell culture in a serum-free environment. Also here, viability is important as well. Something that I would recommend to always use, because you might be lucky, and that is the direct transfer to serum-free media. For many primary cell types like T cells and macrophages, this works perfect, as it does not for some other cell lines. Here an example of which the gradual and the direct approach both worked. In case the direct approach works, this will save you time. So I would always recommend to use both. Often we get the question, are there limitations when moving to serum-free media with regards to the cell types? In general, any cell type should be able to grow in a serum-free environment, taking into account, of course, the weaning protocol, and slowly transferring your cell to a serum-free environment. Well, we talked about the concerns of using serum, the different formulations of serum-free media, and how easy it is to start working with serum-free media. So there are many applications where serum-free media can be used, and all cell culture labs can benefit from working with serum-free media. Just to mention some random examples. So if you would be a researcher working on MAP kinases, some components present in the serum will activate MAP kinases independently of their assay. So that's something you really would like to avoid. Another example, for instance, if you're a researcher working with costly drugs, these drugs can be absorbed by the BSA, the bovine serum albumin, which is present in the serum, and might reduce the effective concentration. So the output that you're getting is off the real concentration. Another example, researchers working with primary immune cells, many mainly human, and they are more and more concerned to use serum containing medium to avoid any artifact caused by serum, because they might stimulate or cause inhibition. So let's look in more detail to some real examples from the market, showing different cell types which were successfully transferred to a serum-free media. Some are published, some are in-house data, and some are kindly provided by our customers. Before I show some examples, I would like to share a short overview of the serum-free media and the Lonza portfolio per cell type. As you can see here are the different cell types, so Chinese cancer ovaria, hematopoietic, hyperdoma, but also some general. And you see that Lonza has different media for different cell type. And this comes back to what I explained to you before, that you can have a protein-free, a non-animal, or a chemically defined media. And that is why we have more choices. So if I give you the example of the CHO, so the ProCHO was the first one on the market. It is a non-animal origin medium, but it does contain some hydrolysates. And then PowerCHO was the second generation, and this was already chemically defined. So the first example is a little bit a special one, because HL1 is not a serum-free media, but I did want to mention it today as it's a product that can replace serum. So instead of adding serum, you add HL1 serum substitute to your classical media. Not in that high concentration as your serum, so for HL1, 1 or 2% is more than enough. And HL1 provides standardization and does not require batch-to-batch testing. Weaning might be required, but most cells adapt easily. It has already been used with many different cell types like lymphoma, cancer, and some primary cells. Some data here where we compared RPMI containing 5% FBS with the same RPMI, but then the FBS replaced with 1% L1 serum substitute. In this case, we worked with a hypersoma cell type and they produce an antibody. So we looked in different weeks to compare the yield of the protein that was produced, which you see in the different colored bars on the left. And we also looked at the microgram IgG per 1 million cells in the same weeks. And what I wanted to share with you, or what would be the message from this, that in this case, HL1 outperformed the serum, but in any case, you can get similar results as you would have with serum by easily going to a serum-free substitute. The first real serum-free media example is ultraculture. And ultraculture is a multi-purpose serum-free media compatible with many different cell types, adherent or in suspension, and it's a complete medium where you only require to add glutamine or ultraglutamine, a stable form of glutamine. To name some cells it's comparable with, it's like with hyperdoma, monocyte, macrophages, epithelial cells, or fibroblasts. So in this case, you can really move away from your classical media with serum to ultraculture. I have to make a small remark here with the ultraculture, if you would be interested immediately. We have at this moment such a high demand for this medium that we are only able to produce the current demand. So if you would like to test it, then start in 2021, because we have looked for some sources to be able to supply increased demand, and that will start in 2021. Just an example here, which is published. So Standen et al., they use Lonza ultraculture serum-free media, which had previously been shown to preserve tumor antigens of primary human glioma cells to be utilized for clinical immunotherapy. And I only show this, I won't go into detail in the publication. You can see the link here. So if you would like to know more how they used it, as this would maybe compare to something that you are doing, I would really advise you to look at the publication yourselves. Moving from general purpose media to cell-specific media. So in this case, insect cells. And of course, they often use the hot topic now for vaccine and of course protein production by use of the baculovirus expression system. So Insect Express is a protein-free medium, and it supports the growth of different cell lines for insect cells. To name a few, SF9, Hi5, SF21, I think there's also an S2, so different ones. And the positive here is that insect cells do not require weaning. So changing to serum-free media is easy. I would always advise to do both, the direct and also the gradual weaning, but for insect cells, the chance of direct transfer is very high. This time, not a comparison with classical media containing serum. This example shows differences in performance between serum-free media for the same insect cells. So even different serum-free media on the market might have a different effect on your cell type as well. In this case, we looked at cell proliferation and protein production, and high yields are of course important when producing a protein. And if you get the same yield earlier, this saves you production time or will increase the total yield. Data that is not shown here, we also did a comparison with a classical media, in this case, Grace media. And of course, this needed serum to grow, and we compared it with our Insect Express. And also here, Insect Express outperformed the Grace media, in this case with regards to the virus titer of the baculovirus. Well, we all know about the hype of CAR T cells. So in this case, T cells, natural killer cells, and macrophages, they become very important cell types for research, and they are used for cell therapy. So of course here high standards are required as patients' lives are at risk. And serum-free media are the medium of choice in this case. Lonza offers 3 different versions. They are named ex vivo, and there's an ex vivo 10, 15, and 20. And ex vivo 15 is the most used media because it's more compatible with natural killer cells, macrophages, and dendritic cells. And we also have ex vivo 10, which is a less rich media, which you use for less quick growing cells. And ex vivo 20 is a very rich media, so if cells grow very quickly, that is the better choice. On request of some customers who had some concerns, some safety concerns when using human transferrin in relation to viruses, I think that specifically came from France. Lonza also has a version containing, sorry, recombinant transferrin, and these are available in ex vivo 15 and 10. So what does ex vivo 15 media mean? So it's a clinical proof of formulation. We have already approved media which customers use in cell therapy, and this helps you in process safety and consistency. Of course, high productivity is important, so a high yield and a viable cell count at the end is important. That is what ex vivo can give you. And of course, it's serum-free, so it's a more defined process, and it limits immune responses from cells. So here a comparison where we used dendritic cells and we compared it with, let's say, classical RPMI containing serum, and we compared it with the two ex vivos that we have, so containing the human and the other one containing the recombinant transferrin. So the serum-free ex vivo media shows better growth performance to serums containing classical media, and there is no difference in cell growth between the media containing human or recombinant transferrin. So with hematopoietic applications, we know that some researchers still add serum to a serum-free intended media, which, to be honest, we do not promote it like that, but well, you know, researchers. So in this example, where they cultivated CD4 and CD8 positive T cells, and they used different media to do a comparison, and they always compared it with and without adding serum. So if you look here at example A, where they have RPMI 1640 with and without serum, of course, we all expected that if you do not add serum, then cells do not grow because they really need the serum to grow. And then B, C, and D are the real serum-free media. And in this case, of course, I would like to highlight ex vivo, where if you add or do not add serum, there is not a lot of difference. In this case, even where you do not add serum, they really need to have serum added. So that's not really, to my idea, a serum-free media. In this case, also a serum-free media where the addition of serum does not have a lot of effect. So for our ex vivo media, it really does not help to add serum. We even have some examples where customers did also add serum and where the serum had a negative effect, so that it had some inhibition on the growth of the cells. This overview shows that there are different media dedicated to specific kidney cells. So we have ultra MDCK specifically for MDCK cells. There's also pro MDCK, that is also a medium for MDCK cells, but in this case it's completely non-animal origin. Adherent, if there is an A, pro 293A, or in suspension, pro 293S. And also for vero cells, which currently are also used in production of vaccines for the COVID situation we currently have. So historically, MDCK cells have been used as models for studying epithelial cells, development and functions. And many studies have shown that MDCK cells are very useful for rapid membrane permeability screening. MDCK in combination with CACO2 are used for these drug transporter assays. HEC293 cells are used for drug permeability studies, and also they can easily be transfected to overexpress some drug transporters. And if you're interested in the use of HEC293 or HEC293 T cells for gene therapy applications for the production of adeno-associated viruses or lentivirus, we have a medium that can support that, and of course, what I already mentioned, that vero cells are also known to be used for vaccine production. Here a comparison, I just want to show this quickly, where also here our pro-MDCK media outperformed a medium, so the DMAM, the green line here, with serum in cell proliferation. Last but not least, in our portfolio, so companies producing recombinant proteins, they often use the Chinese ovarian cells, so better known as CHO cells. And this slides cover the different options of serum-free media for CHO cells. So the difference lies in the formulation and if the media is non-animal origin, protein-free, or chemically defined. So on the left, you see Protein 4 and 5 media. As already mentioned, these are the first one that Lonza had on the market, and they are, of course, of non-animal origin, but they still contain plant hydrolysates. And then we have on the right side the Poucher 1, 2, and ECHO, and these are fully chemically defined media. Here an example when you compare the different media to produce a protein. I have to say, if your protein production, you will always, if you start with a new protein, to do a media screening. So it's always important to test different media with your CHO cell line to see which ones give the best proliferation in combination with the best yield of your protein. Not a serum-free media, but a very important addition to serum-free cell culturing. So once that you have done the effort to wean your cells into your serum-free media, cells should not be frozen in DMSO with serum, which I think is quite a standard way of doing that. As soon as your cell touches serum, then you have to start with the weaning procedure again. So that is why we have on the market ProFreeze, which is a chemically defined freezing media, and this does not require serum. So in this case, that is a supplement to if you have moved to serum-free media. Here you see that there's no difference in viability of the cells kept in ProFreeze or DMSO with serum after thawing. So we compared ProFreeze, 10% serum or 90% serum, and looking at the viability, it's comparable. So not having your cells in serum, but in ProFreeze with DMSO has no effect on the viability of your cells after thawing. I mentioned already that there are several options in serum-free media, like non-animal origin or protein-free or chemically defined or a combination. And in the next slide, I will show an overview which indicates this for the Lonza portfolio, so that you have a good summary overview. I don't know if that's easy to read, but again, here we have all the different cell types, some additional information of the cell types, what they're used for, then the different media of Lonza, and then if they are chemically defined, non-animal origin, animal origin, protein-free, or xenon-free. And for instance, something I did not mention today, but if you look at hybridoma cells, so if you look at ultradoma PF, that means protein-free, it's chemically defined, non-animal origin, and also protein-free. So this gives you an overview if you need to make a choice in a certain media and their application. I would like to highlight our additional sources of information with regards to serum-free cell culturing. Of course, our website contains application pages where we can assess serum-free cell culturing. All our cell-free media with more details can be found on our website at our bioprocess pages. It's also possible to download our specialty media brochure covering our serum-free media. And of course, our weaning protocols, how to adapt your cells to culture in a serum-free environment, are available for download. But you can also ask Biosyn because they also have access to that. You can also have a look at our knowledge centers for specific advice of serum-free media for a specific cell type. And if your cell type is not or does not already recommend a serum-free media, then please get in contact with Lonza scientific support or the scientific support of Biosyn. And if you're interested to start culturing serum-free, the best way is to try it with your own cells. Contact our scientific support or with the product manager of Biosyn to help you suggest which media fits best with your cells. So we are reaching the end of our webinar, and I would like to summarize what we covered today. So we discussed today the concerns of working with serum in relation to standardization, animal welfare, safety, fraud, and future supply. We talked about the serum-free alternatives available on the market, like non-animal origin, protein-free, chemically defined, and xenon-free. I hope I showed you how easy it is by using the weaning protocol to change to a serum-free environment. And I showed different examples of researchers who took the step to go serum-free. I want to end by saying using serum-free media will help animal welfare, and will result in reproducible scientific results. And with this, I would like to hand back to Monica.
Yeah, Nicole, thank you for your very interesting presentation. I think that gives us more confidence to try the serum-free culturing. In Germany, you can contact Biozyme for information. Check out our website, biozyme.com, or contact your local Biozyme sales manager. And now we are open for questions. So you can type them into the chat box. And now we start with the following question. Someone said, maybe I did not get it. Will I need to add 50% of DMSO to the pro-freeze medium? Can I use it for insect cells, for example, high 5?
Okay, thank you, Monica. So let's take them one by one. So do I need to add 15% DMSO to the pro-freeze medium? That's correct. So the final concentration is, so you mix it one on one. So you add 15% DMSO with pro-freeze, so you will end up with a 7.5% DMSO as final concentration. And then you ask, does it work for insect cells? Yes, it works for all cells. So even for cells not in serum-free media, you could use pro-freeze. But for insect cells, yes, for sure it will work.
Okay, now the next question. I do have a question regarding primary murine T cells. What media would you recommend to use, as it is already more difficult to culture those cells than human T cells?
So again, which was the cell type? The mouse T cells.
Murine T cells.
Yeah, that is a challenging one indeed, because our ex vivo media are mainly dedicated for human T cells. But I would think that the ex vivo 15 could work as well. I do not know it by heart. So I see that Alexander asked this question. So what I could do is to check this if we have any references already published with our ex vivo for T cells for marine origin. But I would expect that our ex vivo 15 would be the one that would work, but we will get back to you to be 100% sure.
Okay, so I would say our time is nearly over. I want to thank everybody for joining this webinar, and hopefully you will come to our next webinar in this series. Thanks again, Nicole, and goodbye to everybody.
Thank you. Bye-bye.